So clinical trials are the main mechanism by which we can get drugs for many hematological malignancies, including myeloma. In the development of drugs, so as to get them to use in myeloma to help patients, it is very important to go through the different phases of the trial. First of all, drugs are tested in the laboratory, both in test tubes, so to speak, as well as in animal models. And if they show a signal that they would be useful in a particular malignancy, such as myeloma, they will then be transferred to a phase 1 trial, which often is a first-in-human trial, where the drugs have not been used in humans before. And in that phase, we are mainly concerned about whether the drug is safe and not toxic to individuals, so that they can be brought forward for further development. After the first phase, if it passes, it will then go to a phase 2 trial, where usually in the initial phase, there will be different drug doses if it is shown to be not toxic. And phase 2 often is an expansion, whereby the dose which was found to be effective would be used in a larger number of patients. There are also some phase 2 trials which are already randomized, whereby that dose of a particular drug is compared already with a standard of care, but that is not a full randomized phase 3 trial. Again, it's step by step. So after phase 2, if it passes, it then goes on to phase 3, which often is the most important part of getting a drug to prove that it is not only useful and leads to benefits like improvements in response and survival, but also that it is safe and effective as compared with the standard of care. So in a phase 3 trial, patients, like the throw of a dice, would be so-called randomized. So they would be allocated to either the standard of care, which is the medication which is already being used for patients in that situation, versus the new drug, to see if the new drug actually benefits and improves the results of the original drugs.